Thank you for helping. And welcome to my colleagues again, Tim, who's been with us before. Welcome. Do you want to introduce our guests or shall I?
You go ahead, yeah.
Sure. Well, I'm delighted to introduce Ebony Boulware, the professor in chief of Duke University Division of General Internal Medicine in the Department of Medicine, the director of the Clinical and Translational Science Institute, and the vice dean of Translational Science at Duke University. And my friend and colleague for whom I've been most delinquent, Lola Fashoyin-Aje, Deputy Division Director, the Office of Oncologic Diseases at the Division of Oncology, and the Associate Director, Food and Drug Administration, Science and Policy to Address Health Disparities in the Oncology Center of Excellence. Really wonderful to greet you both. Tim is an old hand at this. I just wanted to start by giving each of you a few minutes to sort of give your perspectives on how we advance this general effort, how we really look to accountability for making change. And Lola, I'm going to come back to you about the recent guidance that came out, I think yesterday maybe, from the FDA. So maybe Ebony, you could start.
Yeah, thanks, Barbara. It's a pleasure to be here. Hi, everyone. I wasn't necessarily prepared to make these overall remarks.
Oh, you don't need to if you don't want to. This is your life. So
what I would say, having seen the presentation and thought a lot about this, is that I think one of the biggest challenges that we face in clinical research in general and in trials is how to adapt like an equity mindset, so that everything that we do really employs the principles or thinks about the principles of equity from start to finish. And I think in the case of, for example, clinical trials, often we have a scientific question in mind, and we may be, for example, looking at two investigational drugs or two different types of treatments. And then sort of the equity question seems to creep in sort of as a secondary question, sort of let's look at heterogeneous treatment effects. Let's look at this sort of after the fact. When indeed we may have to think about that, and I think we should think about that sort of up front. And that might really have a lot of impact on how we even think about how we go about the design of our studies, what some of the fundamental questions really are, who we're going to involve in our designs and in our protocol planning. The other thing I would say is that, I want to make a distinction between sort of the diversity question and the equity question, because I think diversity is about making sure that there's diverse voices at the table. And that's very, very important for a number of reasons. We know that, diversity in teams increases team, creativity and perhaps even productivity and perhaps even effectiveness. And we know that we want our studies to reflect the diversity of individuals in our population. The equity question to me is different in that it's about how do we make sure individuals get equitable access, equitable outcomes. And equity is more about resources, sort of the equitable distribution of resources across groups based on a number of different factors. And I guess I just wanted to make that distinction. I think it's a very important conversation to be having in science. And because in some ways, as I thought a little bit about the presentation, and I had the opportunity to see it, prior to the session, I thought about what it means and why we do sort of heterogeneous treatment effects questions. And I thought about what that should mean in terms of why we are posing a question about heterogeneous treatment effects. Is it that we're truly looking for a heterogeneous effect, meaning that we think, for example, that Black individuals have some sort of different biology than non-Black individuals? In some cases, that could be the case, and there could be a somewhat, and I say somewhat because I really question in many cases that there is any validity to that aspect of inquiry, because I don't believe that fundamentally human beings are different biologically. We know obviously of some clear, genetically driven diseases, but by and large, when we're talking about many conditions, we're not necessarily talking about racially driven differences. When we look at the inequities, we're talking about socially driven differences when we talk about the inequity. And if that's the question, then we might really think about how we design our studies completely differently. Is it a mode of delivery that's really the issue? Is it the access to the treatment that's really the issue? So I guess my broader comments are about really, as we advance it, that yes, it's very important that we begin to think about, and we continue to think about, the diversity of our studies, but also as we start to use the equity term, we really start to think thoughtfully about what that means in this context, and really clarifying those two aspects. That's
a great point. We've sort of talked about it in prior lectures, and it's great to reemphasize now that they're really different frames, and couldn't agree more that this is rarely biology, and wherein there is race differences or ethnicity differences, usually that is, a different factor that is the operative factor, obviously.
And if I could just emphasize and agree strongly with Ebony's point about this, I think that so much of what we talk about when we talk about equity and diversity is that we need to have Black people in our studies. And that's true, of course, we need to have diversity, but your point is exactly right. And the point that that was made earlier in Barbara's talk is that what we really are doing is not trying to treat groups, we're trying to treat patients in front of us. And if we think of it with that lens, that we're not trying to treat women, or we're not trying to treat Hispanic people, we're trying to treat patients by doing this research, then we really need to focus on the factors that you just described. And I would go further to say we need to be thinking about comorbidities. We need to be thinking about age. We need to be thinking about the things that might matter in the treatment of an individual. And having diverse populations will help you achieve that. But it's not the goal to treat by race or by gender. That's not the goal of this at all. It shouldn't be. And there are, I totally agree with you, we shouldn't be thinking about treatments for Black people, because that doesn't mean anything. So why should we be designing studies that specifically stratify by something that we would never use in a treatment, or probably shouldn't use anyway? So I just want to applaud you for those statements, because I think that's something that we often miss. And there is a question here, is there a need in the trial design to stratify by these subgroups? And I think that goes exactly to the point you made. Substratification may not be feasible in the sense of power and things like that, but it also may be missing the point of how we end up treating people if we stratify by things that aren't relevant, like race, perhaps. So anyway, just my 2 cents.
We don't even collect social determinants of health most of the time.
Right.
So. Lola, your thoughts?
Thank you for the invitation to participate in this. That was a very interesting and thoughtful presentation. I think that really summarized a lot of the different perspectives on this topic. I think, I view this issue in a slightly different way, in that I think we do spend a lot of time talking about the limitations of the way that we collect information and data that helps us characterize the safety and effectiveness of treatments. On the issue of race and ethnicity, and whether they're real, biologically based, social, political constructs, I think we've spent a lot of time discussing these issues. And I think sometimes we get lost in those discussions. And while it's important to really kind of define why we're doing what we do, I do think the goal, at least from my perspective, as someone who's reviewing drug development programs to approve new therapies, the goal of all of this is really just to ensure that we have the heterogeneity of the population that will optimize and promote efficiency in our research study, and that will inform treatment of a broader population at the end of the day. And I think, you know, we have you know, clinical and demographic factors that help us begin to assemble that population in a study that is maybe reflective of a general population, but there's imperfection in all of this, right? And none of these parameters or attributes are necessarily correlate with outcomes. We may have some sense of their relationship to the final outcome. It may be a direct one, it may not be a direct one. They may be good proxies or very imperfect proxies. I think we're all in agreement that race and ethnicity are not perfect in sort of helping us necessarily define a genetically diverse population, right? Which I think is kind of what we would need would be really ancestry, which we don't have a good way to collect at this time. But I think there's no doubt that different populations respond to treatments in a variable way. And we don't always know what is driving the difference. And I think when you have a diverse population in the study, you really increase the opportunities to uncover potentially biologic factors that are not related to race, but that may be more enriched in certain groups that have certain genetic characteristics, for example. So I think I don't spend a lot of time talking about this because I think, you know, people know that race is not biology. But I think it's, you know, I think it's not a bad idea to constantly kind of clarify that position. We have seen in some oncology drugs that certain biomarkers are enriched in certain populations. They're not exclusive to those populations, but they're certainly enriched. So if I want to do a study of a drug that is targeting that particular biomarker, this is probably where I need to go, right? And so I think that there's a lot of benefit to including that. And the other point I want to make is that, you know, the way that we evaluate new treatments isn't just based on sort of their biological effects, right? There are other things that impact outcomes. So when we're collecting patient-reported outcomes, for example, those may be informed by things that have nothing to do with biology, but they're not any less important, right? So, and they may be infused with cultural, environmental factors, socioeconomic factors, social determinants of health, and ultimately they may impact outcomes of the treatments. So I say that to just say that I think we should always recognize the imperfections of the way that we are enrolling patients in the trials and in the way that we are trying to get that variability into the trial. There are certain things that are not because of purely biology, but we also can't be dismissive, I think, of some of these efforts because they're not perfect, because we have such a deficit of data to sometimes support our beliefs that you know, I think it's important to kind of look at this from a place of not arguing whether you should have more Blacks in the studies, more Latinos in the studies, but I think arguing from a place of why not? Why isn't that the baseline, right? Like, why are we arguing about whether we should include these people because this thing that we are using to identify them is not a perfect thing, rather than like that's who has the disease and that's who should be studied. It doesn't matter, you know, if it's the biologically sound reason to include them, but like why do we even have to think about including them? It just should be that way. And so I think that's kind of the perspective that I would share, or at least another perspective that I would add to this. Thank you.
Yeah, I love that. I'm an epidemiologist. And so epidemiologists like variation. If we had no variation in our studies, we would never find anything if everybody was exactly the same. And the reason that we can identify differences and identify factors that predict outcomes or predict response to treatment is because people vary. And so the key thing in my mind is to get as much variation as possible. And that could be social access, it could be comorbidities, it could be genetic variability, but the variation is going to teach us things. And if we limit our data and our sample sets to a very narrow range, we'll miss important factors. But that's different to me than saying the goal is to create subgroup specific treatments or whatever. It's to say that by having maximal variability, we'll be able to identify those factors that matter, and then we'll be able to go back to them and tailor treatments or target treatments to those things. And maybe that will be race, but probably it won't be. It'll probably be something else that's maybe correlated with race or correlated with the groups that have high rates or bad outcomes from disease. The one exception, as I think you raised, might be the pharmacogenetics, because if there's actually a biological metabolic event that's happening because of something that's in genetics, that may determine response or toxicities differently. But again, that's not going to be race specific. That's going to be perhaps enriched in some racial groups or other groups, but it'll be found in everybody. So if we can use this natural variation to understand what that pharmacogenetic variation is, and then apply it to everybody who carries that variant, then that's the answer. So your point's just exactly right, I think.
Yeah, I mean, and there's some variants that are more common in certain populations. And maybe it's not race, it's genetic ancestry. We don't collect that data. And so when you have tests, like in the era of precision medicine, right? We have tests that are testing this variant versus that variant, and they're going on these panels. And so but you're not necessarily, these panels don't necessarily include variants that are more common in certain groups. I think that's a problem. And I think that's where I think it's really important to ensure that, you know, we are optimizing the opportunities for us to really be able to identify these different ways that drug response and drug effects can be studied, evaluated, and understood, so that, you know, post-approval, you know, patients are treated, that one patient is getting as much of the benefit as is possible. And I think this also even goes beyond kind of where the treatments have been developed. It's kind of like when you're selecting treatments, you know, there may be treatments that are selected based on how common you're seeing this particular mutation or this particular thing. Again, it's not race-based, but there may be differences in the population. So even at the candidate drug selection stage, I think there are opportunities there to study more people from different parts of the world who are impacted differently by their environment, by their culture, by their ancestry. And I think that's the point of all of this, I think.
I guess what I would say is, just to underscore, I agree with the points of having as much diversity as possible in these studies. I do think that we should not let the perfect be the enemy of the good. However, I do believe that there are some fundamental ways that we have traditionally thought about subgroup analyses that limit our capacity to take advantage of the diversity in the population. So that when we say you need to, how many Hispanic individuals, I'm just taking the NIH language, you know, are you recruiting? How many Black or African American are you recruiting? And then you've met your recruitment criteria. How many women, how many men? And you've met your criterion for diversity just by filling out that table. We're severely limiting the number of axes of diversity that we see within a given individual. So, you know, a treatment in the real world effectiveness may have a different effectiveness based on that individual's capacity to even carry it out. So if I have, you know, 30% of my Black population is low income, and then I have 10% of my non-Black population having low income, and the fact that I have more income in one group enhances my capabilities to take the medication as prescribed, the effectiveness will be different. So therefore, income probably should be one of my axes of diversity that I would include. And ultimately, my hope is that we can get to the multi-dimensional capture of individuals' characteristics so that we can enrich with fully diverse populations. So my kind of point about the diversity with regard to that is not to let perfect be the enemy of the good, but I think to be very careful and cognizant about the types of categories that we do use when we are thinking that we're achieving diversity. We just need to be careful about what that might do and how that might constrain our hypotheses.
So let me push on that for a second. Which is to say, right now, we're not collecting in most drug development trials that are regulated trials for the FDA, for instance, a lot of the variables that you just mentioned, as far as I know. And, you know, and in certain countries, quote, it's illegal, I don't believe that, but it's illegal to collect such information, or so we've heard. And it seems to me that in order to do, you know, we can't require for at least pre-approval of investigational drugs, which is the universe that I think Lola's focusing on, the kind of huge numbers of people to inform that. We either have to say, okay, we're going to learn a lot about that in real world evidence or in phase 4 and post-approval trials, or we have to say we need to really rethink our, you know, statistics and our way of thinking about it, because if you account for every element in the dimensions of diversity, which I completely concur with, you're going to end up with an N of 1 in 10,000 boxes.
Yep.
And it's, you know, when we started the projects we did at MRCT Center, we got a lot of pushback from the pharma companies who said, you know, this is not biology, this is social justice. And I'm like, it's social justice, but until you have the diversity, you can't tell, you know, where there are outliers or what. You know, you leave science on the table by not doing this, which I think is part of Lola's position in terms of you can't know what you don't know until you ask the questions or collect the data. So it seems to me we sort of need to.
I mean, I guess what I would say about that, a couple of thoughts come to mind. First off, pre-approvals, we're talking primarily about safety, you know, and I think we would then be looking for a harm in a particular group. And I just want to we would want to make sure that that would be, you know, that there would be valid hypotheses around harms for particular groups, for safety, drug safety or investigational, you know, whatever it is, safety. I think there's another reason we might want to think about larger groups that have to do with the sort of the acceptability or the trustworthiness of the new investigational agent. So a good example is like the COVID-19 vaccines, where initially when testing was, you know, not among this diverse populations, you had a tremendous amount of mistrust from, you know, from marginalized populations that said, look, I'll wait till, you know, this hasn't been tested in people like me. So I think there can be benefit to having groupings that are socially based that we pull in. But I just wanted to make you know, that distinction again between the biological risks you know, and you obviously need to get past a certain point till you can get those thousands and thousands of participants in there to do the larger studies. And then sort of what are some of these upfront considerations? And it may be useful to, I'm not saying we should not use these groupings, but when we use them, if we're not going to have, first off, large enough groups to make the safety designation anyway, then there may be alternative reasons why we decide to use these larger groups, because we say, look, it has been tested in diverse groups of these particular things, and this, therefore, we may be able to get further, you know, people to participate in future trials where we really need to demonstrate. You know what I'm saying? I think there are these they're very difficult issues to,
Yeah.
and I do think they require a rethinking to some extent about how we deal with these issues.
Agreed. Lola, do you want to comment on effectiveness versus efficacy? Sort of what your goals are in order to say something is approvable? What does sufficient evidence look like? And mention perhaps.
I mean, it's a very complex, you know, thing that considers obviously the clinical setting that we're talking about, but our regulations require that pharmaceutical sponsors demonstrate effectiveness of their drug in adequate and well-controlled trials prior to approval. And that demonstration of effectiveness and the approval pathway can vary depending on the disease, the rarity of the disease, you know, the types of trials that can be conducted, and so forth. But, you know, so there's a requirement for demonstrating effectiveness, and there's also a requirement for demonstrating safety. So that's what needs to be done before a drug is approved. And I think, you know, my general view sometimes is that we do spend, you know, I don't have the luxury when I'm reviewing these trials at the FDA to even think of a world where subgroup analyses by race are possible. That just isn't ever possible, because I have like 5 Black patients and, you know, 2 Hispanic patients, and, you know, so so this is something that I don't run into this problem. I would never advocate for, you know, stratifying by race or ethnicity or any, you know, non-biologically based characteristic. So to me, when we say, you know, enrolling patients, you know, on the basis of their membership to any kind of social, political group of any sort, has a lot of problems. I completely acknowledge that that is not the optimal way to do science. I totally agree with that. But what is the alternative? The alternative is where we are today, talking about how the way we're doing clinical research needs to change. And I think, you know, why have we all bought into this idea that it needs to change? And the reason is because we know that there's a large swath of the population that is not included or represented in our current research, you know, enterprise. And how do we get those people in the trial so we can learn a little bit about them and figure out like what's important and what's not important? I think that's what gets me going every day, is just saying, you know, yes, you know, your skin color is not important at the end of the day. It is for some products, right? Sunscreen and other things. But you know, maybe it doesn't matter as much at the end of the day for a cancer treatment, but I need to get you in a trial. Like, why aren't you in these trials? So I think I'll just stop with that.
Which speaks to case about equity. Go ahead.
Could I follow up on that? To both Lola and Ebony? One of the things that I see in many trials is that there are exclusions made that are unnecessary or seem unnecessary to me, and they exclude people. and they particularly exclude certain minority populations. And you can think about what those exclusions in the trial are. And, they might be comorbidities, or they might be things that are going on that are, common in certain groups of people, and then they get excluded. And so my question is really, is there a way? And I think the reason people are doing that is because they think they're going to get their drug approved more easily if their study is cleaner, so to speak, and it excludes people with things that might cloud the result of the study, or cloud the FDA review. But I'd love to hear your thoughts about whether it's possible, desirable, and how one would go about eliminating some of those seemingly unnecessary exclusions to allow a broader population to participate. Is that a barrier to FDA approval if you have a broader population that might have, disease states that would ordinarily be excluded?
So I think, yeah, thank you for that question. It's one that is very quite often debated here at FDA. Do we approve if it's narrowly studied, or do we hold it for, and it shows effectiveness? Do we approve it and then ask them to study more people? Do we ask them to study more people at the outset? I think we have to have a little bit of a culture shift. And I can say this in oncology because there's tremendous drug development ongoing at the moment in oncology, where, for many diseases, patients have a lot of options. So we're not quite where we were 10, 20 years ago, right, in terms of options. And I think we have to start asking more of these drugs, right? So if you develop a drug where the patient needs to have basically no other problem with them other than cancer, I think that's a problem, particularly if most of the people who develop that cancer have many other problems. And so if you need to have 12 pages of inclusion and exclusion criteria to select your study population to show your marginal drug effect, that's that study population is the healthiest population that will ever receive that treatment. So we have to really develop better drugs, or at least do a better job at being able to predict who is benefiting from the treatment, so that we are not exposing so many people to toxic placebo. So I think from the FDA perspective, we have been involved with many other groups, ASCO, the Association of American Society of Clinical Oncology, Friends of Cancer Research, in really discussing kind of what the scientific and clinical rationale of eligibility criteria are. And we've written many guidances so far, sort of saying, we need to be very thoughtful and select criteria that are clinically and scientifically sound, and not just copy paste, not just coming from a conservative place where you just want to really control that population. Because I think over time, as more options become available to patients, we're going to need to understand that these drugs actually work in the broad population for which they tend to be approved. So that's one thing. At the FDA, we started an initiative where we want to take this efforts around broadening eligibility criteria a step further, and that is in saying, do we need 30 eligibility and eligibility criteria? Are they like a critical set in a particular disease that are all that you need to select who comes into the trial? Because the more pages of eligibility criteria you have, the higher the likelihood that you are going to not be screened into that trial. So that's I think that's the culture shift that we need to have, which is to say, we're not designing patients aren't here to do the studies. The studies are here to develop the drugs for the patients. And that's a culture shift that I think is we're driving there. But I think it's really problematic when people think, oh, well, you can come in my trial and you can't come in my trial. I think the trials really need to be like, who needs the drug, and so let's design a trial around them. So that's the perspective that I share and that we share in oncology at FDA.
Ebony?
Yeah, and I would just say, knowing that, multimorbidity, is inequitably distributed across our population, when we do have these highly restrictive eligibility criterion, we're then saying that we're targeting this towards specific, stratum of our population. So we will tend to have less diverse populations when we do that. So I applaud efforts to open trials up to be more pragmatic, more pragmatically oriented from the start, more effectiveness oriented from the start.
Yeah. And do you want to mention the new guidance that came out, for which comments are,
Yes,
open?
I want to mention it. It's been
Oh,
a
good.
labor of love of mine.
It sure has.
Yeah, so yesterday we published the guidance on diversity plans, and it's essentially a guidance for industry that provides a framework to developers of drugs, biologics, and medical devices, a framework for developing a strategy to enroll a diverse population defined by race and ethnicity in clinical trials and clinical studies. It doesn't provide prescriptive recommendations on specific measures to take, but rather it really sort of describes how to develop this plan that you're going to discuss with FDA early in clinical development, and most certainly before initiating any trials that would support an approval of a new drug or device. It's really simple. And we are going to be asking sponsors to submit these plans. I think that there's a lot of ongoing proposals within Congress around issues around diversity, equity, and clinical trial, clinical research. There are many proposals that are floating about. We have been working on this guidance for quite some time now. And so we're really happy for it to be out, and we are really excited to get people's feedback on it, and to incorporate that and hopefully finalize it in short order. But I encourage you to check it out.
Yeah. I mean, it's of course we're all delighted it's out there, because, you've had in guidance a request for diversity, or an invitation for diversity plans, as it were, but no real expectation in the way that it was interpreted.
Yes, that's true. We published a guidance in 2016 where we had one line that said, we recommend that you develop a plan and discuss it early with FDA. And as I was a relatively young reviewer at the FDA at the time, and I was looking at it and I was thinking, what does this mean? Like, what am I supposed to be? How am I discussing this with sponsors? And so we did a pilot program asking sponsors to tell us their plans. Tell us your plan. And the response was less than.
Well, it's interesting. At the MRCT Center, we sort of worked with a number, with the FDA, obviously, but with a number of pharma sponsors as well as academics. But the pharma sponsors, we read them that line and said, if you could just de-identify, all of the information on those plans, we'll put together a mock, diversity plan. What should be in it? What should people think about? How to approach it? Nobody either had them or shared them, and then we realized big problem. So we developed our own poor man's effort to that. But Tim, one of the things you've done at the Dana-Farber, I think recently, is introduce, for investigator-initiated protocols, a link to the Massachusetts Department of Public Health cancer statistics, so at least people know what our population looks like. Is that fairly stated?
Yeah, I think that, one of the things that we don't, and there's actually a comment in the Q&A about this, that, we don't necessarily try to adapt our studies to reflect the need of specific groups. So the comment in the chat is about myeloma, and so obviously if myeloma is an incredibly higher risk disease in Blacks than it is in other populations, then Blacks should be more better represented in these studies, and that's a really good point. And one way to think about doing that is to understand where these disparities exist. And so yeah, we've started we and others, many people have put together data to try to help understand in some defined catchment area or in a clinical set of the population what those risks look like, and maybe try to help people be guided a little further about what their study populations should actually look like. And again, the FDA guidance might help that, should help that. But the data, people should be looking at the data and trying to understand where these disparities are so that their study populations reflect the general population better.
It really seems to me this is something that, we should have nationally to understand, it's not breast cancer, it's triple negative breast cancer that's, metastatic at presentation or whatever.
Right. Well, and CDER does quite a bit of that. There are data out there, but they're not easily available to people. Somebody who's designing a trial that has to go into the CDER database and come up with these things, that's a pretty high bar for some people. So I think that it's also accessibility, but it should be an expectation that people find this information and reflect it in their studies.
So let me pivot to sort of what does good look like and how do we get there? Because how do we report this? How do we get to a place where, as a society, we really are on one page about, not just getting there, but learning what matters in getting there? Thoughts?
I think optimally, what good looks like is based upon kind of your hypothesis that you're trying to investigate. Like what is the question? What is the study question? Yeah. And I think even if we enrolled in many cases, and I think you alluded to this, Ebony, even if you enrolled a population defined by race that reflects the epidemiology of the disease, for example, you still may not have any numbers to do anything other than potentially hypothesis generating data, right? At the end of the day, potentially. So there may not be, so what is good depends on what do you know a priori, and kind of what is the overall question you're trying to ask. This guidance isn't going to give us data that all of a sudden tells us across all the racial and ethnic populations, this is how these groups respond to treatments. I think we've all agreed that that is not what we're driving at. I think what we want to do is have kind of a consistent efforts to generate data across as variable a population as we can come up with. And maybe those data can be used in pooled fashion or some other ways to identify differences across the population that are not race-based, but maybe based on other things that we didn't think about before we generated the trial. Mm-hmm. So it's not about having the number isn't the goal. The goal is getting your study population to look like the population with the disease. Yep. That's the goal. It's not about, is it 5 patients? Because I get these questions, when I meet with sponsors. Dr. Fashoyin-Aje, when you say adequate representation, do you mean 5 patients, 10 patients, 6 patients? Can they be women? Can they love cats? And it's like, that's not the point, The point is, who has multiple myeloma? Design your trial so we get those people in the trial. Yeah. And let's see what happens.
Yeah.
Um,
And cite the trials where you can get those patients.
exactly.
Ebony?
Yeah, I would completely agree with that. And I would just say, I think the attention needs to be spent from the start of the hypothesis development of the study, with, clear rationale as to, what does the population look like? Why, who do we need to recruit? Why do we need to recruit? And what are those expected? How are we going to go about it? How are we going to, carefully think about how we're going to go about it? What are our expected outcomes with regard to the, the recruitment, retention, enrollment, retention, the process? And then what do we expect to get at the end? Are there any specific diversity-oriented hypotheses that we're designing this for? Or is it an equity question? But I think being very clear on that from the start, and that requires a lot of thought, more than we've been doing traditionally.
Yeah.
Yeah, I mean, a lot of times, we've seen in some classes of drugs in oncology, sort of differences in safety profiles, or, across a population defined by age.
Yeah.
Even though those patients met the same eligibility criteria to be in those trials by renal function or the organ function, bone marrow function, but we still see differences in safety. Now, when these patients were enrolled in those trials, that was not the hypothesis. The hypothesis wasn't, oh, let's enroll a broad range of ages, and because we're going to see differences. These are after the fact things. Now, if you didn't include those patients in the trials, you enrolled all 40-year-olds, you wouldn't be able to kind of do this, say, across all of these drugs, when we look at this subgroup, that wasn't powered, but when we look at these subgroups, we are seeing this consistent trend across the board. Let's design a study studying them specifically to see what comes up. And I think that's how we need to approach this whole thing, is put the people with the disease in the trials and see what happens. But put them in there, put them in the trials. You'll never know what's important or what's not important, or what is the reason why you're seeing differences. I think ideally we would love everybody to respond similarly to the treatment, because that would make people much better, but that doesn't happen.
Yeah.
So
it's interesting, because when as a treating physician, it's important to know that the patient will respond or not, but every patient responds differently, and we know that, and we should be looking to understand that, and we can only do that if we really advance this cause, as you have so beautifully described today. So I just want to thank you for a really educational, informative, and really breathtaking, way of thinking about committing to this effort in a way that makes social justice sense, and grounded in science and biology and understanding. So thank you so much.
And if I can just thank Barbara and the MRCT group. This is the last of the 6 sessions that they just masterfully put together. If you haven't seen all 6, you should go back and watch them, because they are just amazing. There's so much knowledge and so much experience, and this group is just, they've done an incredible job, and I just want to congratulate them for a really amazing series, and I know it's going to have a big impact on lots of people. So thank you, Barbara. Thank you to your team.
Thanks. And thanks for the partnership. And mainly thanks to the participants in this seminar who care to stick with it on Thursday night.
Whenever.
Whenever. And thank you, Ebony and Lola. This has been really wonderful.
Thank you.
Thank you all.
Great. So we're still live.